1, 2, 3. Well, it's again my pleasure to give this lecture as part of the regional training course on wheat blast in Bangladesh. And the purpose of this presentation is to give an update on the epidemiology and management of the emerging wheat blast disease, which is considered an emerging global threat. But before I start, I would like to mention that I am representing the BLAST Integrated Project led by Dr. Barbara Valent at Kansas State University. And this project, for those of you that do not know, the purpose of the project is to develop novel strategies for managing blast diseases on rice and wheat. And today I am going to be talking about one specific objective, which is objective number 3, that has to do with blast control through understanding pathogen genetics, pathology, ecology, and epidemiology. So over the last few years, we have established a good partnership with scientists at The Ohio State University and the USDA, as well as with Growers Association in Bolivia. And the idea is to develop all these strategies that eventually can be implemented by the growers. So we know that wheat blast is one of the most fearsome and intractable wheat diseases in recent decades, according to CIMMYT. So there are many reasons why this is true. So first of all, wheat is one of the most important crops globally with respect to food security. We know that wheat blast strikes the wheat ear and produces sterile heads very rapidly. Unfortunately, the ecology and epidemiology of the wheat blast disease are poorly understood, and fungicides are reported to be unreliable when weather conditions favor the disease. Finally, few resistance genes have been identified. However, we are working right now on identifying novel sources of resistance for the wheat blast disease. So here I have a couple of pictures showing the devastation at the top picture, the devastation caused by this disease. At the bottom, we have affected seeds and also the conidia of this pathogen. So when I started this project, the purpose of my specific project was to assess the vulnerability of U.S. wheat production to blast. And so there were two important objectives as part of these studies. So the first one was to assess the climate suitability for the Magnaportha oryzae triticum pathotype establishment and wheat blast outbreaks in the U.S. And the second objective was to assess the susceptibility of elite U.S. wheat cultivars. And over time, we have been able to expand these specific objectives to more general objectives that could, in the end, help the growers that have been affected not only in the U.S., but also in other countries such as Bolivia, and hopefully we can also be able to help the growers in Southeast Asia. Now, as part of this study in climate suitability for Magnaportha oryzae triticum in the U.S. And establishment and outbreaks occurrence in the U.S., we have developed a model. And basically, the model consists of specific parameters that we have decided based on the conditions that we can find in the United States. So, for example, one important parameter that we consider here is overwintering, that probably is not going to be as important in other parts of the world where more tropical conditions are present. But in any case, there are other parameters that can also be applied throughout the world. So we have, for example, inoculum buildup prior to heading. We know that head infection is probably the most visible and most important type of infection that could occur throughout the plant, because this type of infection directly affects the grain that we want to harvest. However, we have to remember that leaf infection can also occur, and we are trying to determine at this point how important those leaf infections are during the whole epidemiology of the wheat blast disease. Now, the importance here is to determine how that inoculum buildup occurs, and also how infection occurs, but most importantly, what type of conditions are present, and if those conditions that favor the development of the disease can be present in this part of the world. Now, we know that the triticum pathotype is not subject to extreme low temperatures in Brazil, and Brazil is important in this case because we had extrapolated information of wheat blast development in Brazil, where wheat blast is currently endemic. And so because of that, we have relied and we have made extrapolations for conditions of the Lolium pathotype of Magnaporta oryzine, that is a phylogenetic relative of the Triticum pathotype, which is already established in the United States. And so we have considered that freezing temperatures appear to limit the survival of the Lolium pathotype based on previous information that it has already been published. In this case, there is a paper from Harmon and Latin that was published in 2005. And so that information was critical because based on that we were able to make extrapolations and identify those areas in the United States where the pathogen may be able to survive. In this case, this map of the US shows those white areas where both the Lolium and maybe the Triticum pathotype would be able to survive. And so based on these models that we have developed and the assumptions made, when we have run all these models together, we have been able to identify those areas at risk of wheat blast development in the United States. And so, but I'm not going to go into much detail about this, but all I can say here is that we have been able to identify specific areas in the country where it's likely that grain has been received from overseas, especially from South America, and we have estimated the climate risk associated to the development of wheat blast after introduction, and here we're talking about establishment and weather conditions for outbreaks in those specific areas of the country. But again, I'm not going to go into detail today. So what is important about that study is that we were able to identify those key epidemiological gaps, and over the last few years we have been working on trying to fill up those gaps. So, for example, we know that the Triticum pathotype is a seed-borne pathogen, and we also know that heads are affected, and head infection is the most visible symptom in the field, especially when these infected heads develop these bleach color several probably days or even weeks after infection has occurred. So in trying to fill up these epidemiological gaps, we have identified that under certain conditions, the lower leaves or those basal leaves can be affected. And in this case, where I'm showing some seedlings that have shown both signs and symptoms of wheat blast. In this case, we have received this report from scientists in Bolivia. Javier Toledo has shared pictures, and also Guillermo Barea. They both have been working on wheat blast extensively over the years, and they have reported that even the flag leaves under certain conditions can be affected and can show signs and symptoms of wheat blast. So in one study that we had established in the field in Bolivia, we have observed after collecting leaves from those basal parts of the plant, we have been able to identify conidia of Magnaportha oryzae, and in many cases we have been able to retrieve hundreds of thousands of spores in small amounts of those basal leaves. So we have talked already about the presence of those conidia in small amounts of dry leaf matter, and depending on the cultivar that was used and from where those basal leaves were collected, we were able to find significant differences in the amount of conidia found on those leaves. For example, cultivar Atlax, we know that is highly susceptible at the head stage for wheat blast, and in this study it turns out to be the cultivar with the most numbers of conidia on those basal leaves. Hundreds of thousands of spores were found. In comparisons, we found that other cultivars that are more moderately resistant to blast at the head stage were also somehow, let's say, or were showing less numbers of conidia compared to cultivar Atlax. In another study, we had planted highly infected seeds on these bags, and sampling occurred right at the heading stage. And what we were able to find here in this study was that spores were also retrieved and were found on those basal leaves. And whether the source of inoculum was the seed or spores coming from somewhere else, we don't know that part. We're still working on that issue, but at this point, what we can report is that those basal leaves were the ones showing signs and symptoms of Magnaportha infections. Now, on a different experiment under controlled conditions in the US, we had selected 5 isolates and 4 cultivars showing different levels of susceptibility to blast. And those cultivars were inoculated at the seedling stage, and we ran a split plot experiment. And basically, the idea was to determine if there were differences according to the leaf that was infected. In other words, whether the basal leaves were more susceptible to newer leaves showing up in the upper canopies. So the results from this experiment showed that there was a different pattern of reactions across leaves according to the isolate used, but significantly more lesions were found on the oldest than on the youngest leaf when inoculated with one specific isolate that was collected in the 1980s. And significantly more disease was found on the oldest compared to the youngest leaf. In this case, on figure C, that shows the different isolates with that were inoculated to a specific cultivar. All right. Now, what we also reported on this paper was that questions remain as to the main source of spore inoculum. And inoculum produced within the wheat crop was considered that may play a role in wheat blast outbreaks, and that's something that we should consider for future studies. And finally, the epidemiological role of basal leaf conidia is still unknown, and explanation through mechanistic knowledge is yet to be confirmed. So in order to do that, we have partnered with different institutions and researchers in South America and also in North America. And the idea is to keep filling the key epidemiological gaps that we still have. So Gary Peterson and other scientists also, Pierce Paul, we have Carasai Mills, and Diego Valdelomar from Bolivia. We are working right now in trying to assess and evaluate the different data that we have collected so far. So Dr. Larry Madden from the Ohio State University is part of the committee of Carasai Mills, who is a student at the Ohio State University right now. And I almost forgot, Jorge Salgado, a postdoctoral researcher, has also been very helpful during these studies. Now, the idea here is to determine on whether there is an increase in the amount of inoculum over time, and in many cases over space. So we know that inoculum buildup is very important, but the question is whether the inoculum is produced within the fields that are planted to wheat. And so in order to partially answer that question, we have established a preliminary study where we had put infested residue in some treatments, and other treatments didn't receive any infested residue. So we have also used untreated seeds and treated seeds with 2 different fungicides. So the idea here was to collect samples over time and to determine not only the severity of the disease on the different leaves that were present at different times, but also determine and detect the presence of the pathogen and quantify that presence. So here I'm going to be showing the quantification of the presence of conidia on a per leaf basis. So we are still analyzing these data, but what I can present now is that on the x-axis we have the leaf position going from the right side, the flag leaf, all the way to the left side, the F-6 leaf. On the y-axis we have the number of conidia on a per leaf basis, and on the z-axis we have the plot number, which consists of plots having the residue and other plots do not have the residue. This is still raw data and raw results, but I think it's worth discussing them. So in this case, 15 days after emergence of the seedling, we have detected that there is a presence of the conidia on these different leaves, especially on the F-6 leaf. But over time, 23 days after emergence, we see that there is somehow a pattern showing up. So the F-6 showing more presence of the conidia on that leaf compared to other leaves, for example, the F-3. 29 days after emergence, we have found the same pattern, F-6, showing much more conidia on that leaf versus the F-3 or the F-2, which is just showing the presence of the fungus. 43 days after emergence, there's still a pattern. We don't have the F-6 leaf anymore because in this case it has already decayed, and it was really hard to sample that leaf that was already part of the soil in many cases. The flag leaf has shown up, and there is a tendency to have more conidia, or to find more conidia on the lowest parts of the plant. And 59 days after, the flag leaf is showing a good increment on the presence of those conidia. So the question at this point is how we can analyze the data, and how we can use a model in order to characterize the spread of the disease over time throughout the canopy. And so Pierce Paul, among other scientists, have already developed a model that is being applied in the analysis in botanical epidemiology. So we have partnered with Pierce Paul, and he will be in the near future be helping us with using this model to determine the different results that we can get from our data sets. On a different study, Karasai Mills, a PhD student from the Ohio State University, established different plots in Bolivia during 2015, which is considered one of the worst years for blast development in Bolivia. It was a wet year, somehow with mild temperatures going to warmer temperatures towards the end of the wheat season. And Karasai was able to collect data in field hotspots, and she is right now analyzing that data that at some point is going to give us an idea on how fast the disease is spreading over time and space. Now, we also have to talk about disease progress in terms of blast development. So in this case, we're thinking theoretically on how the disease could spread, especially or be present or show up on those affected heads. So we know that the heads are the ones that are showing the most visible symptoms, and so it's worth analyzing how fast the disease would develop in those heads in any given field. But at this point, what we know is that the disease is quick at spreading and at showing up on those heads. And for example, if we give an approximate of 0.1% severity for a practical limit of detection, so from that point on, we only have a very short window to control that disease. We have probably less than a couple of weeks, less than 10 days maybe. And that's the main challenge that growers have, which make me think that if we are detecting the disease at a practical limit of detection, in many cases, maybe that's too late. And what we need to think is what is happening before we are able to practically detect that disease in the field. So in other words, we need to determine what is the main source of inoculum and whether the inoculum is being increased within the wheat fields. So that's why determining the importance of leaf infection is going to be critical to develop new strategies to control the wheat blast disease. In this other figure that was shared by Biotrigogenetica, we're looking at the spread of the disease several days after inoculation. So these are experiments under controlled conditions. On the x-axis, we have the number of days after inoculation from 0 to 16, and on the y-axis we have the disease severity from 0 to 100%. So from this experiment, what I can tell you at this point is that there is one particular cultivar that we are very interested in determining the source of resistance, and that cultivar is ANS 120, which is going to be released in 2017 in Bolivia by the Wheat Growers Associations and Apple. Another important aspect in these epidemiological studies is the rapid detection of symptoms over time. And that is the reason why at this point we're trying to develop phenomics tools to rapidly detect these symptoms. This is going to take some time, but we are considering very highly these type of ideas and technologies. So we have talked about assessing the climate suitability for MOT establishment and wheat blast outbreaks, and now it's time to talk about assessing the susceptibility of elite U.S. wheat cultivars and also identifying novel sources of resistance to wheat blast. So when we started these experiments, we had to determine at what stage we had to conduct the experiments, either at the head stage or at the seedling leaf phase. We know that seedling leaves experiments are quicker and are more practical than head experiments. But after running an experiment and determining the correlation between these two type of reactions at the seedling leaf or head stage, we decided to conduct all of our experiments at the head stage because although there was a positive correlation, the correlation wasn't perfect. And what I'm showing here is the continuous reaction of 85 wheat cultivars that were tested a few years ago. So when we inoculated an isolate that was collected in the 1980s, we were able to identify some cultivars with good levels of resistance. And it turns out that those cultivars with less than 10% head blast severity, from those 22 possess the 2NS translocation from Agilops ventricosa. So we know that this translocation from Agilops ventricosa basically is a 24 to 38 centimorgan segment of chromosome arm 2NS from Agilops ventricosa that was translocated to wheat chromosome arm 2AS. And so this translocation is important, and it looks that is widely available in many cultivars because it possesses different genes that provides resistance. So we have, for example, RKN3, we have LR37, SR38, and YR17. So showing some of the growth chamber and greenhouse results that we obtained from this experiment. In this case, I'm showing some winter and spring wheats and also some near-isogenic lines that were spring wheats that possess the 2NS translocation. In this case, from all those cultivars that were included that possess the 2NS translocation, we found a mean close to 20% head blast severity. But when we tested some 169 winter wheats, the disease severity levels had increased or had doubled to the amount previously found with cultivars possessing the 2NS translocation. And when we included 175 spring wheats with the parents of the near-isogenic lines without the 2NS translocation, that disease severity had significantly increased to close to 75%. So The next question that we had was whether those popular cultivars in South America already possess the 2NS translocation. And so in this case, we had used a newer isolate that was collected in 2012, which is isolate B71. And we had collected the most popular cultivars that are used by the growers especially in Bolivia. And so the idea here was to determine not only the level of resistance to blast, but also the presence or absence of the 2NS translocation. And it turns out that all of these cultivars showing less than 20% are 2NS based. So the conclusions from this experiment, or from this paper, so the first one, the 2NS chromosome segment from Aegilops ventricosa was associated with significant reductions in head blast. Reductions were seen with older and newer isolates and under natural epidemic conditions in the field. However, not all cultivars and lines with 2NS showed resistance in controlled inoculations in the greenhouse. So this suggests that the genetic background or the environment may influence expression of the resistance conferred by the 2NS translocation. So therefore, additional sources of resistance should be identified. So in August of 2016, I assumed the role of BIP objective number 4, team leader, due to the retirement of the previous team leader, Dr. Bill Baucus, and under the new role, I have been able to interact with different institutions and different researchers, not only from South America, but also from Southeast Asia. And I was invited to present some of our results last year during the meeting that was held in Nepal. So after that meeting, we discussed the importance of establishing a protocol that could be used in different countries under different conditions. So, but after coming back from the trip in Bangladesh, we sat with different team members here at K-State and developed this protocol to standardize the inoculation of wheat cultivars that were tested for reaction to head blast caused by Magna Porta oryzae. So this protocol is already available and you can find it online. Now, under these protocols, and especially we need to, or we have pointed out that control conditions experiments are important, but also it is very important to test these wheats or these selected wheats under natural epidemic conditions. And so we have also been working with partners in Bolivia to test not only winter wheats but also spring wheats under natural epidemic conditions. And that is what we are currently doing. We have right now hundreds of lines being tested under field conditions in a location that is considered a hotspot for wheat blast development in Bolivia. And so the idea here is to retest those cultivars that have been selected, especially those having novel reactions or novel sources possible to wheat blast. So to summarize what we have discussed today, wheat blast is an emerging global threat. It strikes the ear and produces sterile heads very rapidly. Although key epidemiological gaps exist, we're working on filling up those key epidemiological gaps. We know that fungicides can be unreliable when weather conditions favor the disease, and few resistance genes have been identified. We have learned today that wheat is important in the US and also in other parts of the world, and therefore vulnerability assessments are very important. In the case of the US, risk areas for establishment and outbreaks exist in the contiguous US, but it's going to be very important to run similar type of studies to determine the risk areas in different parts of the world. Filling ecological and epidemiological gaps is very important. We are working on that, but we definitely are going to need more help in filling those gaps. And definitely identifying novel sources of resistance is very important. What is next in our side? So we are trying to determine the importance of autoinfection versus alloinfection on the wheat blast pathosystem. The development of explanatory or predictive models of wheat blast progress is going to be very important. The impact of climate change on this emerging threat is going to definitely be an important topic to study in the future. And we are currently trying to develop strategies for epidemiological studies using UAVs. In terms of management, it is going to be very important to develop better strategies for fungicide protection. We need to identify non-2NS candidates under controlled and field conditions. We have to identify novel regions associated with blast resistance, deploy 2NS in combination with novel genes or QTLs. And finally, I would like to acknowledge the work done by different people that have worked with us. So in this case, we have Javier Quiyuna from Bolivia. Javier helped us establishing experiments and collecting data for ecological and epidemiological studies. And he has currently joined Kansas State University and is going to be working in the development of PCR protocol to detect Magnaportha oryzae triticum on wheat seed lots. We also have PhD student Giovanna Krupp. She is currently identifying new sources of wheat blast resistance, and she is also interested in understanding the basic ecology of Magnaportha oryzae triticum. And with that, I would like to acknowledge again all of those individuals that have helped us and that continue helping us with our studies. And I would also like to acknowledge the USDA Department of Agriculture, National Institute of Food and Agriculture for providing the funds that have enabled us to continue working with wheat blast. And with that, I would like to thank you all for attending to this workshop. And if anybody has any question, I will be available online to answer any question. Thank you very much.